Hi everyone, this is our first lab in microbiology, and we're going to be talking about lab safety. Now, you guys are taking lab online, so there's a lot of things that obviously don't apply to you because you're doing this virtually. However, you are still required to know all of the safety protocols for any standard lab and what you would be expected to do in the labs on our campus. So the average adult has about 100 trillion human cells and about 1,500 trillion microbes. So you have more microbes in and on your body than you do cells. So you only make up 10% of you, your cells do. The rest of you is microbes. So really and truly, we're all just petri dishes with shoes. So some general safety procedures. Never, ever, ever eat or drink in a lab. Doesn't matter if you have the cap on it, still shouldn't come in the lab. There are things in there that you could accidentally, inadvertently contaminate with and end up really sick. Never apply makeup or put your contact lenses in lab. So I wear contact lenses as well, and if something fuzzy, funny thing floats through and I need to take my contact lens out, I have to leave to do that. Some general safety procedures. We always wear our PPE, which stands for personal protective equipment. In our labs, this would be a lab coat, gloves, goggles, and always, always closed-toed shoes. That means shoes that have a closed heel and do not have any openings. So no sandals, no Crocs, no anything like that. Your feet should be completely covered. All right. You don't have to look like the guy in the first picture, but you do have to look like this. All right. So you want to make sure that you have all of the personal protective equipment on when you are in the lab. If you leave the lab, you take all that off, you come back, you put all that back on. So you don't wear your PPE out in the hallway as well. We always tie long hair back. Last thing you want to do is end up with your hair full of microbes. Before each lab period, you should read over the scheduled exercises so that you know what you're doing. Place coats, extraneous books, purses, backpacks on the racks in the back of the room. Some colleges and labs don't even allow you to bring those in. They have lockers outside of the lab for you to put things, including your books, all those kind of things. There was a situation up at Clemson a number of years ago where inadvertently a tube of, I think it was strep, was spilled on someone's lab manual, and then they took it back to their dorm and they got everybody really, really sick. And so they have super strict rules up there about what you can take into and out of microbiology labs. So you always want to be careful. Don't bring anything extra that you don't need because there's microbes everywhere in there, and we deal with some pretty nasty stuff in our labs that can make you very sick. So don't bring anything extra in. Some safety procedures we want to know. What are microbes? That's what we're talking about this whole semester. So we're talking about bacteria, fungi, viruses, and protozoa. In our teaching labs, we deal primarily with bacteria, but in other labs that you may come in line with, they would maybe deal with some of these other microorganisms. So some more microbiology safety procedures we want to be aware of. Why do we need special handling for microbes? Because you're thinking to yourself, well, they're all over me already. Why do I need to be extra careful in the micro lab? Some microbes can cause infectious diseases. We deal with some pretty nasty stuff in the labs. And if you're working in a hospital medical laboratory, you could be dealing with some really nasty stuff. So we always want to keep that forethought in our mind. Special protocols must be implemented to minimize the risk of laboratory acquired infections, what are called LAIs, to students, faculty, staff, and their families. So an LAI is any infection acquired through the laboratory work or through the lab related activities, regardless of whether they are symptomatic or asymptomatic in nature. So you could easily carry something home to your family that may not make you sick, but it may make another family member uh, that has maybe a less robust immune system very sick. So we always want to be aware of that. Okay, so what types of microbes can be handled in our microbiology lab? The Centers for Disease Control is going to divide microbes into 4 biosafety levels based on their ability to cause disease. And so that is posted, what level lab we are dealing with is posted on the outside of the door. So the biosafety level designation of a lab is based on the highest biosafety level of organisms used, and this in turn determines the biosafety practices that are used by those working in that lab. Only organisms designated BSL 1 and 2 are used in our microbiology teaching labs. We are designated on campus as a biosafety level 2 lab. So what does all of that mean? A biosafety level 1 is the least restrictive. The organisms that are worked with in that lab are not known to cause any disease in healthy humans. So things like staph epidermidis, which is all over your body right now. Biosafety level 2 means that it's a moderate risk organisms that are being used in that lab that are associated with less serious human diseases. The potential for transmission is limited. There are proven treatments for any diseases that could be manifested from something you came in contact with in lab. Some examples of this are Staphylococcus aureus and Streptococcus pyogenes, or the causative agent of strep throat. The upper levels, 3 and 4, those are only going to be in research labs, and so and medical labs, so you're not going to see those on campus. Some other things to know: we wash our hands when we enter and when we leave the microbiology lab. This was always a source of great frustration to my students if they needed to leave when I was still teaching on campus, to go to the restroom or anything like that, because you had to completely disassemble yourself, take off all of your PPE, and wash your hands to leave, and then you had to wash your hands when you came back. Even if you just washed your hands in the bathroom, you have to wash your hands and assemble all of your PPE. So I used to always say, make sure you pee before you go to class, because leaving is kind of a big ordeal. We want to clean our lab bench with disinfectant at the beginning and the end of each lab session. I'll be talking a little bit later in the semester about the appropriate amount of time that different disinfectants have to be left on a surface. This is good information for your home too. But every product that you use to disinfect, it has instructions, and we really should probably read those instructions, because it tells you how long you need to leave the product on there. So the different products that we use in lab, the instructor would say, you need to spray the lab, and then you need to let this sit for 2 minutes or 1 minute or 5 minutes or whatever the instructions on the label say before you actually clean it up. This whole idea of cleaning the lab bench is going to protect you and prevents contamination of your cultures. So we always assume that the lab bench is dirty when we come in, even though we know that the people that were there before us cleaned it up. At least we hope they did. Um, so we always assume that. So we always clean it when we come in, and we always clean it when we leave. Some other things to know: we don't place contaminated loops, needles, or pipettes. So any instrumentation that we use to move microbes around, or to manipulate microbes, are not put on that lab bench. We incinerate the loops and the needles to sterilize them, and we dispose of pipettes in the designated receptacles. If we have to carry test tubes in the lab, and we do this a lot because you'll have a specimen and you need to go get it and you need to bring it back to your bench, and then you need to take it and dispose of it. We never carry test tubes around in with just our hands. We always use a test tube rack. There's less risk that you will drop it this way. If you drop the whole rack, that's a different deal, but there's less possibility that you're going to drop or spill a rack than you are an individual test tube. Okay, so where do things go when we need to dispose of them? We have three choices: the everyday trash can, the biohazard can, which is what they call the red bin. So it is got a special bag in it and it is red. And then a broken glass box, which in most labs is blue. And it's a cardboard box that you put certain things in that have broken. So if we're talking about used plastic petri dishes, they're going to go in the biohazard can. So they're going to go in the red bin. What about used plastic pipettes? Same thing, because they are contaminated. We don't want to put them in the regular trash can. We want to put them in the biohazard can where they're going to be safely disposed of. Live tubed cultures, again, in the biohazard can. Used test tubes are going to go in what's called the broken glass box. All right, in that broken glass box. Used cotton swabs, biohazard. Used glass slides in the broken glassware box. Used paper towels can go in the trash can. Why? Because why you used them was to mop up disinfectant. And so they are already disinfected. So they can go into the regular trash can, and used gloves also need to go into the biohazard can. Food and microbiology labs don't mix. As I mentioned earlier, absolutely no food or drinks are allowed in the lab, even if it is capped, even if it's in a container. Any of those things, you should never, ever, ever bring anything that's going to go into your mouth into the lab. Additionally, no cell phones, tablets, or laptops are to be used in the microbiology lab. Infectious bacteria can hitch a ride on these and then you take them home. So we don't use any of those things in our lab. The exception to this is sometimes cell phones. So I often ask students in lab to use their cell phone to take a picture through the microscope so that they can use it in a report. And if that's the case, we make sure that we put on clean gloves to do that. We get out our phone, we disinfect everything to before we do that procedure. But I do sometimes ask that. But in general, your cell phone is not out in lab. All right, just a some quick data about infections that happened in clinical and teaching microbiology labs in the past, the human Salmonella typhimurium infections in particular. In 2011, there were 109 cases in 38 states. In 2014, there were 41 cases, multi-state. And in 2017, there were 24 cases in 16 states. I'd like to draw your attention to the states that we are in right now, and that is South Carolina. And you can see there was a limited number there, but I unfortunately live in Georgia. And look at the number that were in Georgia. Now, I'm going to guess it's because the CDC is in Atlanta. And that's why you see that bigger number there. But nonetheless, it is kind of an interesting thing to look at. In all of these outbreaks, behaviors such as not wearing gloves or lab coats, lack of hand washing, contaminated items such as cell phones, pens, and the like were being used and taken out in lab and associated with infections. So you might be saying to yourself, well, we don't use that particular species of salmonella in our lab, so why does this matter? What it does is demonstrate that students can become infected with organisms used in the microbiology lab if they don't follow biosafety guidelines. We use commercially acquired low-risk organisms to reduce the likelihood of getting sick. That does not mean that you won't get sick. So you are required to follow biosafety guidelines to reduce the exposure to organisms in our teaching labs. Bet all that makes you really glad you're taking this online, right? So what you work with can make you sick. So make sure you follow safe lab practices and don't bring germs home with you. Always wash your hands with soap and water right after working in your lab, just before you leave the lab. All right, so we wash our hands. You become hand washing fanatics when you take microbiology. You want to avoid contamination in the lab. You don't eat or drink, as we've talked about, or put things in your mouth like gum or candy or anything like that. Nothing goes in your mouth while you're in the lab. You don't touch your mouth or your eyes, because that's a great way to introduce pathogens um, to your body. And don't put on a cosmetics. The big one I always remind people of is lip balm, because people don't think about lip balm as being a cosmetic, but it is. So apply that, and make sure you don't handle your contact lenses. Like I said, if something happens to your contact, you always want to walk out of the room after you wash your hands. Don't carry dangerous germs from the laboratory home with you. Leave your personal items outside of the lab so that they don't contaminate your cell phone, your car keys, your tablet or laptop, any other type of devices. Keep work items off the bench area when you do experiments. So your backpacks, your notebooks, your pencils, your pens, all those kind of things can't be sitting up there on that lab bench where you're working, and you leave your lab supplies inside of the lab. So you leave everything that belongs to the lab in the lab. You don't get to take anything home with you. And so you don't take your lab coat home with you. You don't do any of that. You leave all of that there. So leave your experiment inside the lab so you can stay healthy outside of the lab. Critically important, report any accidents to the instructor immediately. Okay, so if you spill something or you break something, it is not a big deal. All right, nobody's going to yell at you or take away points or do anything crazy like that. We just want to make sure that everyone is safe. And so if you spill a vial of pathogen or a vial of microbes, just let someone know. Okay, make sure that someone is the instructor and that it's cleaned up properly. All right, so you have 2 virtual labs in Connect to do, one on hand washing and one on personal safety. Again, we're doing our labs virtually, and you guys are not setting foot in a lab for this particular course, but you will go into labs in your other courses that you take, assuming you're going into some type of healthcare. So we want to make sure that you know all of the appropriate information to do that successfully. And I will see you for the next lab. Bye.